All right, so we have finished talking about the plasma membrane, and the next organelle I want us to take a look at, and largely the role of the nucleus within the context of development is going to be in the context of gene expression, kind of where the chromosomes are, and how the nucleus is going to facilitate that process. And then, of course, we'll see the nucleus itself participates quite a bit in cell division, and specifically the mitosis and meiosis portion of cell division. So in this section, what we're going to do is talk a little bit more about the nuclear envelope as a structure. So not surprising, and you'll remember back those figures that we used to have in intro books, that the nucleus tends to be the largest, most prominent organelle in the cell, and of course eukaryote cells are named after the fact that they have that nucleus. And we would probably say that the main function in the cell of the nucleus is to hold and protect that genetic material, those chromosomes. There are other events that take place inside of the nucleus, but almost all of them revolve around the presence of those chromosomes. So the outer boundary of the nucleus is a membrane. So this is a membrane-bound organelle, similar to the other ones in the cell. And the outer boundary is what's called the nuclear envelope. And this, of course, is going to separate the nucleus from the rest of the cell, which is called the cytoplasm. And this would include not just the aqueous environment of the inside of the cell, but also all the other organelles, and the plasma membrane would be part of the cytoplasm. This guy is a little bit unique in that it is still a membrane bilayer made out of phospholipids, but in this case the envelope, and this is somewhat indicated by the name, is actually a double bilayer, meaning that the bilayer circles around on itself, so that you actually have it's almost like a bilayer inside of a bilayer. And the other interesting thing about the envelope is it is continuous with the endoplasmic reticulum. So any changes that are made within the endoplasmic reticulum with regards to their membranes will translate directly into the nuclear envelope. The space inside the two layers is what is called the perinuclear space. We won't really worry about that too much because the functions of it are not going to affect our developmental processes to a great extent, but there are times where what's going on in the perinuclear space is going to interact with the inside of the nucleus and have some events take place, similar to, if you remember, the mitochondria has two membranes, an inner membrane and an outer membrane, and there are events that take place across that inner membrane. This sort of inside of the double bilayer has things happen across it in a similar way. All right, the nuclear envelope also is going to have pores. So rather than transporters that would be embedded into the membrane, what happens is the double bilayer kind of folds around and creates these pores, and inside those pores are what's going to be called a nuclear pore complex. It's a large structure. You can see them basically under the light microscope if you have high enough power. And this complex is going to essentially regulate what comes in and out of the nucleus. Just a reminder here, we'll kind of discuss this in a little more detail later, but the cytoplasm and the cytosol, this is one of those things that at this point you definitely need to be able to tell the difference on. So the inclination at this point is to call the inside of the cell and that liquid portion the cytoplasm. Cytoplasm is actually everything. Cytosol would actually be the fluid portion. So cytosol would be the environment, aqueous environment inside of the cell that's going to interact with the aqueous environment inside the nucleus as things pass back and forth to each other. We'll talk a little bit more about the inside of the nucleus in a little bit. This is what it basically looks like. So the gray bars here are actually the phospholipid bilayer. So that thing would have two leaflets. You'll notice that again it's continuous with the ER. So the ER would basically circle around and probably extend out and about. And you can see the double bilayer comes around and tucks under. This would be the perinuclear space. These would be the pores. So again, you can see how the phospholipid bilayer comes, tucks under, and continues through the underlayment, or the inner layer of the membrane, and basically creates this sphere with the interior of this nucleus, and of course out here would be the cytosol where we would interact. Here's another look at it, kind of zooming in on the nuclear pore, and you can also see the double bilayer here. So here would be one bilayer with its hydrophobic core, here's the other bilayer with its hydrophobic core, and this would be the space in between and the nuclear interior. So this would be the membrane that some things happen across as we move molecules back and forth, or atoms back and forth. And then the nuclear pore complex here is where anything goes through. And notice anything that's going to go through is going to go all the way to the interior or come all the way out to the cytosol. And here's a electron microscope look at them. So you can see the lamina here, the envelope as it lays on top of the lamina, and then these would be the nuclear pore complexes. And you can see how many of them there are. Basically the nucleus is just covered with these nuclear pores, and this would be the lamina underneath that you see. So in that picture, the nuclear envelope itself has been removed, so you can see the structures left behind. Within the nuclear pore complex, so again, this is what regulates what comes in and out. So there's two transport mechanisms that I want you to be familiar with in this case, and there are essentially two, well, they're sort of related to each other, so it's really one big cargo or transport mechanism, and depending on the direction that your cargo is moving, you will tap into a set of transporter proteins. And the importance, as you might guess, are going to recognize things that have a nuclear localization signal, meaning that I'm supposed to go in the nucleus, so carry me over there and put me inside. And then exportins, of course, are the reverse. They pick up things inside the nucleus, carry them outside of the, through the nuclear pore complex into the cytosol, where they will then do their jobs. So here's a good summary slide to kind of show it. So nuclear localization signal on your cargo, importance bind together, those two come inside through the nuclear pore complex. Once the cargo is delivered, this guy goes back outside to pick up somebody else, and then of course the reverse is here. Nuclear export signal picked up by exportin, sends across, drops it off, comes back inside. And I guess actually we should go this way. Comes back inside, picks up something else. And these guys will cycle back and forth across this nuclear pore complex, importing and exporting all kinds of materials. So it's estimated that in something like a human liver cell, that hundreds of thousands of molecules are transported across these nuclear pore complexes every minute. So a lot of activity going on with these import and export mechanisms. Here's an interesting experiment to show the effects of those nuclear localization signals and nuclear export signals. So here is a protein that has its nuclear localization signal in place, and it's been loaded with a fluorescent dye. And so you can definitely see that under normal conditions, these guys will be imported inside. And then if you take the nuclear localization signal away and put it in the cell, you'll notice it actually gets stuck out in the cytoplasm, so it is not able to move inside. Whereas here the reverse, if you put a protein here that does not have an export signal, then it gets stuck inside. But if you add a nuclear export signal on it, then those proteins will come outside. So the export signal is critical for this to happen, and localization signal is critical for this to happen, and we basically get the movement of things inside and outside the cell. So what kinds of things get transported in and out? It varies depending on the cell type, but these would be things that certainly would be common. Any kinds of enzymes that work inside of the nucleus need to get in there and do their jobs for things like transcription, DNA replication, and other reactions that have to take place. Structural proteins, which we'll talk about when we talk about chromosomes in the next section. Gene regulators, transcription factors are going to have to get inside. Things that come out would be things like mRNAs, ribosome subunits, and all kinds of tRNAs and small RNAs that also do other jobs. So a lot of transport taking place, even when we're sleeping. So basically this is a process that never shuts down inside all of our cells. All right, so next time what we'll do is dig into the inside of the nucleus, look at the chromosomes as well, and that should tell us what we need to know about the nucleus. Bye for now.